Okay, we'll wait for 2 minutes for letting other people join and then we'll start the session. Okay, so we'll start the session. A very good evening to one and all present here. Welcome to the week 4 online discussion forum. So today we'll be covering all the topics, all the lectures that were covered in the week 4 of RNA biology. So in week 4 there were 3 lectures, and one by one we'll cover each lecture. We'll just have a quick uh, revision of what all points were covered in all the respective lectures, and then we'll solve the questions. Okay, so let us start. So week 4 was about RNA processing and life cycle. Okay. So in the previous week, in the last week, we have studied about transcription. How transcription happens in the case of prokaryotes and how transcription happens in the case of eukaryotes. So both prokaryotic and eukaryotic transcription we have covered. And we saw that there are the what are the main 3 steps which are involved in RNA transcription. The first one is initiation, second is elongation, and the third is termination. Initiation involves the formation of the first peptide, first bond. Okay. And then when the first bond is formed, then um, the transcription bubble moves along the DNA, and this movement of transcription bubble along the DNA is your elongation step. And finally, when the RNA has completely made, then the transcription has to stop, and that is known as your termination, transcription termination. Today we'll see that what are the various post transcriptional modifications, or what are the various processing of the RNA that occurs after the transcription has taken place. So you need to understand that when your RNA is formed from your DNA, it is in a very naive state. Naive state as in, hmm. So when RNA is formed from the DNA, it is in a very naive state. Naive state means that this RNA, which is formed from the DNA, has to undergo a series of events, a series of processing events in order for it to become completely mature and go on for the next step of the cycle, which is translation. So, for the translation to occur, the RNA has to undergo various processing events. Okay. And when I say that the RNA has to undergo various processing, some post transcriptional modifications, I am essentially talking about eukaryotic. Why? Because in the case of prokaryotes, transcription and translation are a coupled process. Okay. We have already discussed in the last class that transcription and translation are coupled in the case of prokaryotes. So everything is in the cytosol because there is no well-defined nucleus and its boundary. So everything is in the cytosol. So simultaneously, your transcription takes place and your translation also occurs simultaneously. But when we talk about eukaryotic transcription, in the case of eukaryotes, your RNA is first formed in the nucleus, it is processed, and then it comes out of the nucleus and it is in the cytoplasm where the translation occurs. That means, see, the environment of the cytoplasm is reducing. So this reducing environment, plus there are many exonucleases as well as endonucleases that can act on your RNA. And since RNA is very prone to degradation, it is highly possible that your RNA can get degraded when it comes to the cytoplasm. And that is the reason why transcription and translation are coupled in the case of prokaryotes, so that there are lesser chances of the RNA to degrade. Because once you're waiting for the complete RNA to make and then you will start the translation process, there are chances that RNA might degrade. So here in the case of eukaryotes, since first the transcription has to complete and then only it can come out of the nucleus, that means RNA has to make itself in such a way that it is protected from the exonuclease activity which is present in the cytoplasm, and it is also protected so that translation can happen properly. That means RNA has to undergo some processing events, because these processing events will make RNA uh, uh, strong enough so that it can come out of this and it can live in the cytoplasm. Okay, so lecture 1 is about RNA processing. So, the primary transcript which is formed after the transcription take place, it is not the exact transcript which is used for translation. So when I talk about RNA processing, when we talk about RNA processing, we are talking about actually the conversion of the primary transcript into the mature RNA. This is the cytoplasm and this is the nucleus. So your primary transcript is formed in the nucleus, and this primary transcript undergoes certain RNA processing events which ultimately converts this primary transcript into a mature RNA. And once this mature RNA is formed, then only this RNA comes out of the nucleus into the cytoplasm. Fine. So there are three major modifications that happens which converts this primary transcript into a mature mRNA. What are those modifications? First one is the removal of certain nucleotides, addition of nucleotides to the 5' or 3' ends, and modification of certain nucleotides. So when I talk about removal of nucleotides, essentially we're talking about splicing. Okay. Addition of nucleotides to the 5' and 3' end. What are we talking about? 5' capping and 3' poly A tail addition. And modification of certain nucleotides, certain kinds of methylation or demethylation, whatever. So these events converts a primary transcript into a mature RNA, and then only it comes out of the nucleus. So this is what RNA processing is about. RNA processing is the conversion of the primary transcript into a mature RNA. So newly formed RNA molecules, whatever the newly formed primary transcripts are, they undergo various alterations to yield a mature product. And very few RNA molecules are transcribed to final mature RNA. This is important, which means that if suppose there are 100 primary transcripts formed, it doesn't mean that all 100 will be converted to mature RNA. Very few of the primary transcript can successfully undergo all the modifications and then they can convert to mature RNA. Is it clear? Fine. So, three points were there: removal of nucleotides, addition of nucleotides, and modification of certain nucleotides. So removal of nucleotides by both endonucleases and exonucleases. So endonuclease, it will cut within the RNA molecule, and exonuclease will trim the ends of the precursor. So endonuclease will cut within the RNA molecule, whereas exonuclease will cut at the ends. Okay. Then this general process is seen in prokaryotes and eukaryotes for all type of RNA. This is sub, sub it occurs in prokaryotes and eukaryotes both, but certain modifications are specifically in the eukaryotes. Then addition of nucleotide to the 5' end of the primary transcript or their cleavage potent. So you can see at the 5' end, 5' capping occurs, and at the 3' end, poly A tailing occurs. So these points we are going to study in detail later. Okay, don't worry. We are going to study all these points in detail. We are just started, and we are just getting a gist of it. Then modification of certain nucleotides on either the base or the sugar moiety. So addition of the methyl group. So methyl group addition, methylation occurs. Okay. So next is RNPs. So this concept was also discussed in your class. So I have just touched it so that you know what this concept is about. So RNP means ribonucleoproteins, which means they are RNA protein complexes. Okay. And for example, one example of RNP is ribosome, which is the largest and the most complex RNP. So ribosomes are the largest. Ribosomes are the largest and the most complex RNP which are known. So what are ribosomes? These are the biosynthetic machinery for protein synthesis. So they are protein biosynthetic machinery. They are composed of two subunits, 30S and 50S subunits in the case of prokaryotes, and 40S and 60S subunits in the case of eukaryotes. And in the case of prokaryotes, when I talk about prokaryotes, it is known, it is a, what happened? Just a minute. Fine. Yeah. In the case of prokaryotes, okay, I think there is some problem. Let me just try to find out. It will not work, I think, this way. There is some problem. Okay, fine. I'll write it here only. I don't know, there is some kind of problem that's happening. Yeah. Okay. So in the case of prokaryotes, the ribosome is known as 70S. In the case of eukaryotes, it is known as 80S. Okay. And 70S, the subunits are 30S and 50S. And in the case of eukaryotes, 40S, this is 80S, it is 40S and 60S. Now you can say that 30S and 50S, but it doesn't add to 70S, and a 40S and 60S, it doesn't add to 80S. So these subunits are not additive. This S represents the sedimentation coefficient. So this represents the sedimentation coefficient, and these are not additive in nature. Okay. So now here what I was telling you, that prokaryotic ribosome is 70S and it has two subunits, 50S and 30S. 50S subunit again has 23S rRNA, 50S rRNA and 31 proteins, and 30S subunits again has 21 proteins and 16S rRNA. Whereas when we talk about the eukaryotic ribosomal structure, it is much more complex than the prokaryotic ribosomal structure. You have 80S subunit, which is further as 60S, which is composed of 60S and 40S subunits. This 60S subunits has these many components, and 40S subunits has these many components. This is important. Okay. So what are the subunits and what are the components? This is important. So just try to remember it as much as you can. Fine. Then Little bit about the tRNA processing. So we have already studied that RNA is P. Why we are talking about tRNA? Because we are talking about ribonucleoproteins. So ribonucleoproteins are basically RNA and protein complexes, and one of the most widely acceptable example of ribonuclease proteins is your ribosomes. And ribosomes are the largest and the most complex ribonucleoproteins. So ribosomes can basically, the eukaryotic ribosomes and prokaryotic ribosomes are different. How they are different, that we have already just discussed. Then we come to tRNA processing, RNase-P and ribozyme. So tRNA processing in the case of prokaryotes, first we'll see, and then we'll see later in the case of eukaryotes. So tRNA processing in the prokaryotes, how it happens? Mature tRNA are generated by processing longer pre-tRNA transcript, which involves specific exo and endonucleotic cleavage by RNase DEFP, followed by base modification, which are unique to each particular RNA type. So what happens when we talk about the tRNA processing in the case of prokaryotes? You have primary transcript. Now these primary transcripts are acted upon by nucleases, exonuclease and endonucleases both. Okay, and after they are uh, treated with, I mean, they get treated with some kind of exonuclease, then certain kinds of base modification occurs, which converts them into a mature tRNA. Okay, so this exonuclease and endonucleatic activity, uh, which happens with RNAs D, E, F, and P, it's a generalized activity. But to make each particular type of tRNA, this base modification happens. So this base modification actually makes unique types of tRNAs. Okay, this base modification makes different unique types of tRNA. This is what we are talking about tRNA processing in the case of prokaryotes. Now let us see what happens in tRNA processing in the case of eukaryotes. So in the case of eukaryotes, the pre-tRNA is synthesized with a 16 nucleotide leader sequence, 40 nucleotide intron, and 2 extra 3 nucleotides. Okay. So in the case of eukaryotes, in the case of tRNA processing, in the case of eukaryotes, primary transcripts form secondary structures which are recognized by endonucleases, and then 5 prime leader and 3 prime extra nucleotide is removed, and then this enzyme tRNA nucleotidal transferase, it adds this 5 prime to 3 prime CCA, and then the introns are removed. So basically intron removal takes place, and this is important. This third point is important. I'll just mark it as like this. Okay, this third point is important, which says tRNA nucleotidal transferase. Okay, the name of the enzyme is tRNA. This enzyme, what does it do? It adds 5 prime to 3 prime CCA. So this addition actually generate the mature 3 prime end. Okay, and then obviously there is intron removal which takes place. So we are talking about tRNA. So why we are talking about tRNA? Is to show that this is a ribonucleoprotein, which is RNase P. This is actually involved in tRNA processing, and therefore it becomes important. Then, oh, sorry. So what is RNase-P? RNase-P is the RNA component that can catalyze pre-tRNA in vitro in the absence of protein. Thus, RNA-P is a catalytic RNA or a ribozyme. So, if there's a question, name one catalytic RNA, then what would be your answer? The answer would be RNase-P. RNase-P is a catalytic RNA. Okay? And then in the lecture 1, there was this table where it was shown how to read this these triplet codons and how to read that, how these triplet codon codes for which amino acids. Okay, fine. So now we have completed whatever was there. We have just revised very briefly. I have tried to just make a quick notes of what was all there in your lecture one. So I always try key whatever has been dealt in the previous week, whatever lectures were there, I try to summarize them and give you a gist of them. And I also take screenshots of some important points, so that within 2 hours we can complete all the three lectures, and plus we can do the questions. So you're not only doing the questions, you're also revising the portion which has been covered in the previous week, so that like you can do your assignments. I mean, it is helpful for your assignments only. So that is why I try to do it in this way. Fine. So now we'll do just quickly, we'll do one or two questions which are related to this lecture one, and then we'll proceed further. Okay. The question is, rho factor is found in A, plants, B, animals, C, bacteria, or D, fungus. This is your question. I'll give you 2 to 3 minutes. Right now it is 7:23. I'll switch the screen in 7:26, and then you tell me what is the correct answer. You can just write it in the message box. 'Excuse me. 2 more minutes. Okay. 1 more minute. Okay. I'm switching the screen now. Okay. Hmm. Okay. So the correct answer for this question is bacteria. That means rho factor is found in bacteria. Okay. The next question is, sigma factor is a part of DNA polymerase, B, RNA polymerase, C, splicing, or D, mRNA decay. You have 3 minutes to answer this question. Two more minutes left. One more minute left. Okay. So the correct answer is RNA polymerase. How is it RNA polymerase? Let us see. So sigma factor is a part of. So we have already discussed that for transcription to take place, there is an enzyme that is RNA polymerase. Okay. RNA polymerase is the enzyme which is used for the transcription process to take place. Now, this RNA polymerase has different subunits. Uh, and these subunits first makes the core enzyme, which is alpha 2, beta, beta dash. These four makes the core enzyme. And then sigma factor comes and binds to it, and it makes the holoenzyme, which means this sigma factor is a part of this RNA polymerase, and this sigma factor makes the RNA polymerase holoenzyme, and then only this RNA polymerase can initiate the transcription process, because this sigma factor is needed for the recognition of the promoter sequence. So only when the RNA polymerase can recognize the promoter sequence, it can sit on the promoter sequence and it can initiate the transcription process. So for the promoter sequence to be recognized, sigma factor has to come and make this core enzyme, convert this core enzyme into an holoenzyme. Therefore, the correct answer would be RNA polymerase. Okay, fine. Now we'll come to the lecture 2, which is RNA processing and life cycle. Here we are talking about the RNA splicing. So, when we talk about the processing of mRNA, in bacteria, there is no processing of mRNA, and therefore there is rapid degradation at the 5 prime. Why? Because whenever we talk about processing of mRNA, we one of the processing that occurs in mRNA is the 5 prime capping, and this 5 prime capping kind of protects the mRNA from any kind of degradation. Since in bacteria there is no 5 prime capping that happens, therefore in bacteria, in the case of prokaryotes, the mRNA which is formed, the newly formed mRNA, is very much prone to rapid degradation from the 5 prime, and therefore the coupled transcription and translation is important. It becomes important in the case of prokaryotes, because otherwise it is in the cytosol, they are the mRNA is highly prone to degradation. Therefore, coupled transcription and translation is the only way to rescue it. Okay, so just if we look here, eukaryotic mRNA processing ka overview. See, this is your DNA, and one of the strand of the DNA acts as a template, and then transcription happens, and this is your primary transcript which is formed. Now this primary transcript undergoes various modifications. The first modification is the RNA at the 5 prime end, which is RNA, methyl 7 methyl guanosine addition, which is 5 prime capping. So, see, it's not like all the three post transcription modifications happens together. No, there is a sequence in which these post-transcriptional modification happens. So first, your 5' capping takes place, and then your poly A tail addition takes place, and after your 5' cap and poly A addition, then your RNA splicing takes place. Okay, so first is capping, second is your poly A tailing, and third is your RNA splicing. This poly A tailing is helped with the help of the enzyme which is known as poly A polymerase. So this is just an overview of how mRNA processing occurs in the case of eukaryotes. Okay, so now one important topic to understand here before we again come to your this thing splicing is HNRNP and SNRNP. So what is HNRNP? HNRNP is HNRNA plus protein and SNRNP is SNRNA plus protein. Okay, HNRP is HNRNA plus protein and SNRNP is SNRNA plus proteins. Fine. Now, what is the use of this hnRNA and snRNA? This hnRNA, hnRNP, it helps the hnRNA in single-stranded form. It prevents the formation of any unwanted secondary structures. Okay? So basically, your hnRNA has to be in the single-stranded form. So this hnRNP, it kind of helps the hnRNA to be present in the single-stranded form. So how it prevents the hnRNA to remain in the single-stranded form? By preventing the formation of any unwanted secondary structure. So it prevents the formation of any unwanted secondary structure, and therefore it helps in putting the hnRNA in a single-stranded form. Then we come to snRNP. What are snRNP? They are synthesized in the nucleus by RNA polymerase 2 and have a normal 5' cap. Okay. They are exported to the cytoplasm where they associate with the common core proteins and with other specific proteins. And their 5' cap gain two methyl groups and they are then imported back into the nucleus where they function in splicing. So these snRNP are again snRNA and proteins, and they are present in the spliceosome complex. So they help in splicing, define methylation sites in pre. Okay, so these snRNP basically are present in the spliceosome complex, and these snRNP, they helps in splicing. So important point is that first these snRNP are synthesized in the nucleus, then they go into the cytoplasm, and then they gain two methyl groups in their 5' cap, and then they again go back to the nucleus. And when they again go back to the nucleus, then they associate with the spliceosome complex, and then they help in splicing. So basically, this snRNP is required for the splicing process, but for it to help in the splicing process, it itself has to gain some kind of modification so that it can help in the splicing process. Okay. So how it gains those modifications? So it gains those modifications by going to the cytoplasm, gaining two methyl groups in the 5' cap, and then again going back to the nucleus where they help in the splicing complex. With associate with the spliceosome and help in the splicing process. Okay, this is your SNRP. So why we are helping? Why we were reading about this SNRP and HNRP? Because they both are involved in your splicing. They both help in the proper and functional splicing to take place. So what is splicing? Splicing is nothing but the process of cutting off the pre-mRNA to remove the introns and joining together the exons. Okay. So basically, your pre-mRNA, which is Hmm. Pre-mRNA which is formed, it has introns as well as exons because it is formed from the DNA. So DNA has introns and exons both, but RNA should only have introns, sorry, exons, because exons are only mRNA. We're talking about mRNA because exons are only going to um, tell which protein sequence has to be synthesized, which protein has to be incorporated. Therefore, this is exon, intron, exon, intron, exon, intron. Okay. Now, these intronic sequences have to be removed from the mRNA. And then it becomes, suppose if it is E1, E2, and E3, it becomes E1, it becomes E2, it becomes E3. All the exons, all the introns are removed. So this removal of introns is known as splicing. We are splicing out the introns. So it takes place in the nucleus before mature mRNA can be exported to the cytoplasm. So all the post-transcription modifications, first is 5' capping, then poly-A tailing, which takes place with the help of poly-A polymerase, and lastly your splicing. All these RNA processing events, all these modifications happens in the nucleus before this mature mRNA can be exported to the cytoplasm. Okay, so which means that the mRNA which is exported to the cytoplasm is mature because it has undergone all the processing events so that it can withstand the reducing environment and all the endo and exonucleatic activity which is there in the cytoplasm. So these introns are nothing but the non-coding sequences and exons are the coding sequences. And RNA splicing is the removal of introns and joining of exons. And splicing mechanism must be precise enough to maintain the open reading frame, which is catalyzed. And this splicing is catalyzed by spliceosome, which is RNA plus protein complexes. So they here important point they are saying that splicing mechanism must be precise to maintain the open reading frame. Why? Because see here, I'll tell you. If this is your sequence, whatever it is, splicing ATG is the first codon. Okay, is it? Yeah, first codon which codes for AUG. Okay, it is AUG, which will code for methionine, and then GCGU will code for something. Okay, I'll write it fairly. Okay, AUG, CCG, TGA. Okay, suppose this is your codons. Splicing has to be in such a way that first codon, if first codon has to be methionine, then it should be, the frame should be read like this. AUG, then CCG, then TGA. Yeah, I mean it should be U. Okay, it should be U. If splicing happens, if the splicing is not correct and it gets modified somehow and it is not precise, and then if suppose if you are reading the reading frame like this, UGC, CGU, GA, and whatever is there, then your whole reading frame is disturbed in this case here. And ultimately it will lead to incorrect protein or formation. It can also lead to improper protein folding. It can also lead to improper protein folding. Therefore, the splicing mechanism has to be very accurate enough. It has to be very precise enough so that the reading frame is maintained. Okay. Hmm. Now it says that splicing requires a set of specific sequences to be present. Okay. The 5' end of almost all introns has the sequence 5 to 3' GU, and the 3' end is usually 5 to 3' AG. The GUAG sequence, the AG at the 3' is preceded by a pyrimidine rich sequence called the polypyrimidine tract. This is something, I mean, nothing as so it is self explanatory. Nothing to explain that much in this point. Just you have to remember that what are the sequences which are required at the 5' and 3' end during the splicing complex. So now we have completed the lecture 2 also. Quickly we'll do 2-3 questions which are related to lecture 2, and then we'll move to lecture 3, and then we'll do questions of lecture 3. So this week how we are doing is that instead of dealing all the questions at the end and you getting bored of all the lecture that I'm giving you, I have framed it in such a way that I'll cover first lecture, then we'll do the questions which are related to first lecture. Then I'll go to the second lecture, and then we'll cover the questions which are related to second lecture, and subsequently for all the lectures like this only. So that there is a balance between your theory as well as your questions. So I hope this is, I think this is a better way of dealing the theory as well as the questions. Because if I only deal questions, these question deal will not take, I mean, if you're just covering up the questions, it will be over in I think 30 minutes. We have a 2 hours class, so we have to take some theory portion as well, so that it not only complete the time frame, but also more importantly that your concepts are getting revised. And if you have any query, if you have any doubt, you can ask me. This is a great opportunity for you to ask your questions, ask your doubts, and raise your problems here. Okay, fine. Now let us see the question. Question is, enzyme that do not cut RNA is A. RNase, D. bDNase, C. RNA endonuclease, and RNA or RNA exonuclease. So, what is the correct answer? I'll give you 3 minutes. 7:45. I switched the screen. 7:48. Think what can be the correct answer. 2 more minutes. One more minute left. Okay. So the correct answer is DNA. Why? See, such a simple question requires only presence of mind. RNA cuts the RNA. RNA antinuclease will cut somewhere in the interior of the RNA. RNA exonuclease will cut at the extreme ends. And DNA is cut at the DNA. But here we're talking about the enzyme which cut RNA. So RNA will obviously be cut by all these things, but not DNAs, because DNA will act on DNA, not RNA. So the correct answer is DNAs. Okay. Now the next question is, an example of RNA protein complex is tRNA, ribosomes, mRNA, or RNase P. Okay. I'll give you 3 minutes to solve this question. Right now it is 7:49. Okay. I'll switch the screen after 3 minutes. I'll switch the screen in 51. So now this is, see, this question can be a little confusing. But let us see how you try to answer this question. Okay. We'll see. 2 more minutes left. One more minute left. Let us see. Okay. So the correct answer is ribosomes. So example of RNA protein complex is ribosomes. And you see tRNA, mRNA, RNase P. RNase, why it is RNase P can also be an example, but here we're talking about the example. We're not saying what does RNA protein complex means. RNA protein complex is RNase P, and the example of this is ribosomes. Here they are talking about the example, not the meaning of RNA protein complex. RNA protein complex synonym is RNase P, but the example is ribosome. That is why it is ribosome. Okay. So don't get confused over there. Okay. Next question is, tRNA processing is done by RNase H, RNase K, RNase A, or RNase P. So, which of the RNA is required for the processing of the tRNA? This we have studied, I think, in the last class also. We have studied in detail how tRNA processing occurs. And today also in the lecture, we have discussed, I think in the lecture 1, we have discussed how tRNA processing occurs. So this should be clear to you guys. Okay, I'll give you 3 minutes. Right now it is 7:53. I'll switch at 7:56. Okay. 2 more minutes left. One more minute left. Okay, let us see the answer now. Okay. So the question is, tRNA processing is done by RNase H, RNase K, RNase A, or RNase P. And the correct answer is RNase P. We have already discussed that RNase P is required for the processing of the tRNA, and it kind of removes some of the nucleotides and it converts the primary tRNA into a mature tRNA. So the correct answer is RNase P. Okay. So just a minute. Okay, so now we'll come to the topics that were covered in the lecture 3. So in lecture 3 was RNA processing and life cycle, the post-transcriptional processing. So here we are studying about the splicing. So in the previous lecture, we saw that splicing is the process of cutting off the introns and joining of the exons together, and the introns are the non-coding sequences which has to be removed so that the coding sequences which actually codes for the proteins, they are only present in your RNA. So RNA essentially has only the coding sequences. Okay, RNA does not have any non-coding sequences. RNA only has the coding sequences. So this splicing occurs with the help of some kind of machinery, some kind of complex, some kind of enzymes are needed for the splicing to take place. And this machinery is known as a spliceosome complex. This spliceosome complex, it catalyzes the pre-mRNA splicing in the nucleus. And this spliceosome complex is composed of 5 small nuclear RNA which are associated with snRNP. So let us see very quickly. I'll just tell you. Okay. Hmm. Very quickly, I'll tell you. See, this is your exon. This is your exon 1 and this is your exon 2. And in between is your intron. So when splicing happens, this intron kind of folds on it in itself and forms this kind of a structure. And this structure is known as a lariat structure. So this intron is removed from the exon through this lariat structure. And when this lariat structure kind of intron is removed, then these two introns are then joined together. So this is how splicing takes place. Now this is just like an overview. Now let us see how this happens. So actually, if you look on the molecular level, this intron has certain nucleotides which are present at the 5' end of the intron. This is the 5' end of the intron and this is the 3' end of the intron. Okay. So certain nucleotides are present at the 5' end of the intron and at the 3' end of the intron, and A is present somewhere in between the intron, which is known as a branched site. Okay, now GUAG is important and it is conserved and it is needed for the splicing to take place. So what happens? Enzymes, sequential addition of the spliceosome complex enzymes takes place. Okay, there is sequential addition of the enzymes. So what happens? First U1 and U2 comes. Just a minute. Okay. So what happens? First, U1 and U2 comes. U1 come and sit at GU. U2 comes and sit at A, which is the branch site. Okay. Then what happens? U4, U5 and U6 complex, everything comes. And then what happens? You see here, this is the branch point, this is AG and this is GU. So first, this gets cut from here, this goes to the branch point, then again there is a cut over here and then it is removed. So first there is a cut at GU, it gets folded here, and then there is a cut at the 3' side AG, and then the lariat structure is removed. So GUAG. So whenever you are learning about spliceosome complex and the mechanism and processing of how splicing take place, it is important to remember these three things. GU at the 5' end, AG at the 3' end, and at the branch side you have A. So GUAG is typically conserved when we talk about splicing. So this is important. So this is how splicing take place. So intron is removed in the form of a lariat structure. Means first there is a cut from here at the 5' site and then it kind of folds on itself, and where it goes and binds, it binds to the branch point, and then there is a cut here and then this is removed. Okay, and then the two exons are joined together. This is how splicing takes place. Okay, so this is how splicing takes place. So there are three modifications. There are three post-transcriptional modification: 5' capping, 3' poly A tailing, and then splicing. And what is the order? First your 5' capping occurs, then your 3' poly A tailing occurs, and then your splicing occurs. This is the sequence. This is first, this is second, and this is third. Now let us see what is what is 5' capping. This is the first post-transcriptional modification that happens. So very soon after RNA polymerase II starts making a transcript, and before the RNA chain is more than 20 to 30 nucleotides long, the 5' end is chemically modified. So how the 5' end is chemically modified? By the addition of 7-methyl guanosine. And there is a 5' to 5' linkage. These 2 points are important, students. These 2 points are important not only for your this examination point of view, but in general these 2 points are important if you are going for any competitive examinations or anything. These two points becomes important there. So shortly after the initiation process of the transcription, 5 prime capping has to occur. Now what are the functions of 5 prime cap? It protects from degradation, it increases the translational efficiency, it helps in the transport of the mRNA to the cytoplasm, and it helps in the splicing of the first exon. So as I told earlier also, that the cytoplasm is a reducing environment. Okay, so when your mRNA from the nucleus it goes to the cytoplasm, it comes in the reducing environment, and RNA is already very prone to degradation, and in the cytoplasm it is a reducing environment, plus there are so many exonuclease and the nuclease activity are present, so RNA can be easily degraded. So to protect the RNA from degradation, at least from the 5 prime end, what is their 5 prime capping happens. So when this 5 prime capping happens at the 5 prime end, then this RNA becomes resistant to degradation from the 5 prime end. Also, this 5 prime cap actually kind of acts as a signal, and it helps in directing this RNA to the nucleus. See, you need a way, you need a direction, just like we have Google Maps, right? So we have Google Maps, and when we set our destination and we click on Google Maps, on the Google Maps, we know that which direction we have to go. So how RNA knows that from the nucleus where it has to go? It has to be directed. So this direction is helped with the help of this 5 prime cap. Okay, so it is not only protecting the RNA from the degradation, it also helps in the transportation of this RNA to the cytoplasm, and it increases the translational efficiency because your RNA is intact, and it also helps in the splicing of the first exon, because 5 prime capping is the first modification that happens. Okay, so we have covered splicing also, we have covered 5 prime capping also. So we'll be learning about these modifications in detail in subsequent classes also. Here we are just briefly touching it. So for the students who joined late, just a minute. Okay. So this time how we are doing is that we are revising, like for the week 4 there were 3 lectures, right? So how we are doing is that week first we are revising the important concepts which are there in lecture 1, and then we are solving one or two questions which are related to lecture 1. Then we come to lecture 2, we are quickly revising the concepts which are there in lecture 2. Then we are solving one two questions of lecture 2, and then we come to lecture 3, and then this is the lecture 3, I think last lecture, and then we'll quickly revise the see the questions which are related to lecture 3. Okay, so now next is 3 prime cleavage and polyadenylation. So we covered splicing, we covered 5 prime capping, and this modification is 3 prime poly A tail, which means that at the end of the RNA a lot of AAA are added. So therefore we call it as poly A tail. Poly A tail. Poly means many. A means adenosine. Tail means because at the end it is acting as a tail. This is poly A tail. So now what happens is that RNA polymerase 2 does not usually terminate at distinct size. So what happens that if this is your pre-mRNA, first your suppose if this is your polyadenylation signal, downstream to this pre-mRNA is cleaved approximately 20 nucleotides downstream of polyadenylation signal. If this is your polyadenylation signal, okay, this is your polyadenylation signal, then approximately 20 nucleotides after this polyadenylation signal, your pre-mRNA is cleaved. Okay, and then what happens? Many A's are added over here. So like this it happens. Is it clear to everybody what I'm trying to tell? That what happens here is that this is your polyadenylation signal, and approximately 20 nucleotides away from the after the polyadenylation signal there is a cleavage, and then there is the addition of the A adenosine. So now important thing you should know, upstream and downstream. What is upstream and downstream? It is in reference to a point. Suppose if this is my point, upstream means this, downstream means this. Okay, so if this is my +1 site, start site or transcription site, upstream of the transcription start site means before the transcription start site, and downstream of the transcription start site means this way. This is upstream and this is downstream. This is a term, commonly used terminology in molecular biology. So you should know this. Okay. Then we come to other kind of modifications. So three modifications which are the major modifications that happens after the transcription has taken place. What are those? This is 5' capping, then 3' poly A tailing, and splicing. Apart from these methylation modifications, there is one more modification which happens, which is pre-mRNA methylation. So methylation happens after these three modifications occurs. What is methylation? Final processing events in many pre-mRNAs. The methylation patterns are largely conserved in mature mRNAs, and it makes mRNA resistant to degradation. So basically, before this mRNA goes into the nucleus, this happens. This modification, methylation happens, and it makes the mRNA resistant to degradation. Degradation by several nuclease activity. Now, this is what we have covered. Another important topic, which is very much important here, is alternative splicing. Now, what is alternative mRNA processing? So, whatever we have studied up till now are the general, this is very interesting actually, are the general mRNA processing that we know, capping, poly A tailing, and splicing, and all that. Now, see, I'll tell you, it's very interesting if you have not read this mRNA, same mRNA with some exons E1, E2, E3, E4, and so on can give rise to many different proteins. This is one mRNA, okay? But this one mRNA, excuse me, sorry, just a minute. Mm, okay. This one mRNA can give rise to many different proteins. It can give rise to protein number 1, it can give rise to protein number 2, up till it can give rise to, let's say, protein number 11. So how is it possible? mRNA is the same only. How same mRNA can give rise to many different proteins? This is because this mRNA can be processed in different ways, which can give rise to different proteins. Okay, a very new and a very interesting concept. And this is known as alternative mRNA processing. Now, this alternative mRNA processing can happen in three ways: alternative polyasides, alternative splicing, and RNA editing. Let us see what are these. Let us see. So alternative mRNA processing is the conversion of pre-mRNA species into more than one type of mature mRNA. So the pre-mRNA can be, see, we know that pre-mRNA is converted to mature mRNA. Okay. So this one pre-mRNA can give rise to many different kinds of mature mRNA. And this is because of the alternative splicing. So how this happens? Because of alternative splicing and alternative poly A processing. Let us see how it happens. Before I explain from here, I'll try to explain you in a very easy way, very simple and a very easy way. Okay. See, this is your pre-mRNA, which has exon, intron 1, exon 2, intron number 2, exon number 3, intron number 3, exon number 4, intron number 4. Okay, this is your pre-mRNA. Now I will tell you how this pre-mRNA can give rise to more than one kind of mature mRNA. First, I'll take alternative splicing. So now I am splicing intron number 1 and intron number 2. I get E1, E2, E3. Fine. I am splicing only intron number 1. Rest I am not doing anything. So I'll get only E1 and I will get only E2. Or I splice intron number 3 and intron number 2. Then I will get E2, E3 and E4. I will not get even here. This is this is mature mature mRNA number 1. This is mature mRNA number 2. This is mature mRNA number 3. So depending on that which introns I am removing, I will have different combinations of exons. Is it not the case? So which means that differential splicing by different splicing patterns, we can have different set of exons which are joined together. Okay, so this will create different types of mature mRNA. And when different types of mature mRNA are created from this will ultimately give rise to different proteins. Okay, so this means that same mRNA is same pre-mRNA, same primary transcript is giving rise to different kinds, sets or combinations of exons. This is known as alternative splicing. What is it known as? Alternative splicing. Okay, now next one is, so let us see alternative splicing. So the generation of different mature mRNA by using different promoters, using different just by retaining different introns, by retaining or removal of certain exons. So alternative splicing, this is one way of alternative splicing. There are other ways also, but they have made it in a very confusing way. Now we'll see by having different poly A sites. What does it mean? Some pre-mRNA contain more than poly A sites and they may be used under different circumstances to generate different mature mRNAs. How? In one cell, the stronger poly A site is used by default, but in other cell, a factor may prevent using stronger site from being used. So now we'll see the different poly A sites. How different poly A sites can be done? If this is your mRNA, so important thing, we said that mRNA has a poly A site and downstream approximately 20 nucleotides downstream of the poly A site, there is a cut. The cut happens and then there is an addition of AAA with the help of the enzyme poly A polymerase. This is what we have just studied. So important thing is that your mRNA can have more than one poly A site. This is poly A site number 1. Okay. This is poly A site number 2. Fine. This is poly A site number 3. So there are 3 poly A sites. Okay. Now what happens? Let us see what will happen now. If I am cutting here, I will have a different length of mature mRNA. If I cut here, I will have a different length of mature mRNA. If I cut here, I will have a different length of mature mRNA. That means depending on which poly A site I am using, I will not only have a different length of mRNA, mature mRNA, the sequence would also be different. This will have sequences only up till here. This will have this additional sequence also. This will have even more sequences, which means that by using different polyacids, I am producing different types of mature mRNA. Am I not producing? Different polyacid usage will lead to the production of different types and different lengths of your mature mRNA. So this is again your alternative processing of the same primary mRNA. By using, here the concept comes, by using different polyacids. We will be studying about these things in subsequent classes also, but I thought that I'll just give you a gist of how this happens. Today I think in detail I have explained how this alternative splicing and how this, by using different polyacids, it can be done. So now whenever it comes again, you know the concept behind it, because see, just reading out the textual matter will not help you to understand the concept. So I try that whatever conceptual matter is there, you have already gone through in your lectures. If there are some important concepts which I think need some extra attention, I'll try to give more time on those points. Okay. Am I audible? Was I audible? I was audible, no? I just wanted to know that I was audible up till now or not. I hope I was audible to you. Okay, fine, whatever. Okay, thank you. Because this was on mute, I thought, what if I was not audible and I was just speaking for such a long time? Okay, so now we have covered lecture 3's portion also. Quickly, we'll do the questions. And because many of the students joined little late, very in 5 or 10 minutes, I will just briefly revise lecture 1, lecture 2 also. No worries about that. But first, let us complete these questions. Okay. So the first question is, which is not a post-transcriptional event? A. Transcription termination. B. Splicing. C. 5' capping. And 3 is polyadenylation, which is not a post-transcriptional event. So what you do is, what you guys have to do, I'll give you 2 minutes for you to think and answer. You type your answer in the message box. Okay, after 2 minutes, I will switch the screen. I will go through the message box. I will see what you have answered, and then we'll discuss the question. Like this, we always do. So, right now it is 8:22. After 2 minutes, I will switch the screen. You write your answer in the message box. Okay, whatever you think is the correct answer. Try to attempt, try to see, and try to answer it. Try to answer the question. You have 1 minute more left. Then I will switch the screen. Okay. Okay. Now I'm switching the screen. Let us see what you have answered, whether you have answered or not. Okay. So Roshan has answered transcription termination. So, which is not a post-transcription modification? Yes, the correct answer is transcription termination. Why? Because transcription termination is one of the events that occurs during transcription. No, we have already studied about it. That first, what happens? Initiation, then what happens? Elongation, and then termination. So, initiation, initiation, transcription elongation, and transcription termination. These are the three events that occurs during transcription. It comprises transcription. When transcription termination happens, then your transcription is completed. After that, your post transcription modification starts, and your post transcription modifications are 5' capping, polyadenylation, and splicing. So the correct answer is transcription termination. So Roshan, good, you have answered it correctly. Okay, next question is, RNA transcription is in 5' to 3' direction, true or false? 2 minutes to answer this question, although it is a easy question, but okay, I'll give you 2 minutes. Think what can be the answer. Right now it is 8:25. I'll switch at 8:27. I'll switch the screen at 8:27. Okay. 1 minute more left. One minute more left with you guys. Then I will switch the screen. Okay. Time's up. Now I'm switching. See whether you have answered. Okay. So Roshan has answered true. So the question is, RNA transcription takes place in the 5' to 3' direction? Yes, it takes place in the 5' to 3' direction only. So always remember, whether we talk about transcription or we talk about replication, everything take place in the 5' to 3' direction. Okay. Next question is, genes are always transcribed from the non-template strand. True or false? Genes are always transcribed from the non-template strand. True or false? I will switch the screen at 8:29. It is 8:27 right now. Okay. Think what can be the right answer. Okay. I am switching the screen now. Let us see. So again, Roshan has answered at false. So genes are always transcribed from the non-template strand. It is false because any of the strand can be taken for the transcription process to take place. Okay. So this is false statement. Just a minute. I don't know. Yeah. Okay. Okay. Now, next question is RNA polymerase 2 stringently regulated than Pol 1 and Pol 3. Means RNA polymerase 2 is more stringently regulated than RNA polymerase 2 and RNA polymerase 1 and RNA polymerase 3. So for people who don't know, uh, basically when we talk about eukaryotes, in eukaryotes there are different RNA polymerases. Okay, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Why there are different RNA polymerases? There are different RNA polymerases because there are different kinds of RNA which has to be transcribed. There are rRNA, there are mRNA, there are tRNA, there are some snRNA, there are some nuclear RNA, RNA. So different kinds of RNA require different kinds of RNA polymerases. So there are essentially five kinds of RNA polymerases. I think RNA polymerase 4 and 5 are basically involved in plants. So different RNA polymerases are required for the transcription of different types of this thing, RNA. Okay, so now the question here is that RNA polymerase 2 is more stringently regulated than polymerase 1 or polymerase 3. So is it true or false that you have to tell? So right now it is 8:32. I will switch the screen at 8:34. Okay, 1 more minute left. One more minute. Okay, now I'm switching the screen. Hmm. So, okay. So Roshan has answered two. True. That RNA polymerase 2 is more stringently regulated than polymerase 1 and polymerase 3, which is the correct answer. So, yes, RNA polymerase 2 is more stringently regulated because RNA polymerase 2 is involved in the transcription of which RNA? It is involved in the transcription of mRNA. Okay. And basically mRNA is the one which is going to give the codons. It is going to code for proteins. Okay, so whatever the functional product is formed, that is formed from mRNA. So that is why the enzyme which is transcribing mRNA need to be more stringent in its action, because everything depends on RNA polymerase too, right? Because mRNA is going to give you the codons which are ultimately going to decide which amino acid will come. The sequence of mRNA, whatever the sequence of mRNA, A, T, G, 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 sorry, A, U, C, C, G, U, A, whatever it is there, this is going to give you the triplet codons. Is it not so? And these triplet codons are going to decide which amino acid will be incorporated in the protein. Therefore, the enzyme which is going to synthesize mRNA has to be very stringently regulated. Okay. So, this is the answer of this question. So, the next question is, let us see the next question. Yeah, prokaryotic RNA polymerase has only 3 subunits. True or false? Right now it is 8:36. I will switch the screen at 8:38. Okay. One more minute left. One more minute left with you. Okay. So I am switching the screen now. Let us see. I think this is the last question. Yeah, I think it's the last question. Okay. So Roshan has answered false. Yeah. So prokaryotic RNA polymerase has only 3 subunits. Obviously, it is false. Why it is false? Very quickly. See here. So when I talk about RNA polymerase, so the RNA polymerase is different for prokaryote and eukaryote. That we know. In eukaryotes, we have many different subunits also which are present, and I mean subunits. Yes, many different subunits are present, and also in the case of eukaryotes, you have different kinds of RNA polymerase. You have RNA polymerase 1, RNA polymerase 2, 3, and so on. So in the case of eukaryotes, you have different types of RNA polymerase. But the subunits of RNA polymerases are also different. Eukaryotic RNA polymerase is more complex as compared to prokaryotic RNA polymerase. Still, eukaryotic RNA polymerase does not have only 3 subunits. What are the different subunits of a I'm sorry, I think I'm making you confused. Prokaryotic RNA polymerase and eukaryotic RNA polymerase. Okay, this is more complex Okay, it has greater number of subunits, and there are many different types of RNA polymerase. Here it is simpler, but still it does not have only three subunits. What are the subunits that prokaryotic RNA polymerase have? Alpha 2 means two alpha subunits, beta, beta dash, and I think omega is also there. I mean alpha 2, I think alpha 2, beta, beta dash. This makes core enzyme. When sigma factor also comes and join with it, it makes holoenzyme. And this holoenzyme is important because this holoenzyme will only help in the recognition of the promoter. So when RNA, so I have already explained in the last class that to initiate the prokaryotic transcription, sigma factor is required because sigma factor actually recognizes where the promoter is present. So sigma factor goes and sits on the promoter, and then the RNA polymerase recognizes that this is the promoter, and then it starts the initiate the transcription process. So although eukaryotic RNA polymerase is more complex than prokaryotic RNA polymerase, still prokaryotic RNA polymerase does not have just three subunits. Okay, so very quickly I will try to summarize the whatever we have dealt in this lecture today. So RNA processing a life cycle. So what we have studied, that what is the meaning of RNA processing? RNA processing means the conversion of the primary transcript to the mature transcript, and this takes place within the nucleus. Okay, so the conversion of the primary transcript into the mature RNA, the mature transcript is known as RNA processing, and this RNA processing occurs inside the nucleus. After this, after the RNA has matured, only then it comes out of the nucleus. And what are the three essential steps in the conversion of primary transcript to the mature RNA? Removal of certain nucleotides, addition of certain nucleotides to the 3' and 5' end, and modification of certain nucleotides. Removal of nucleotides means we're talking about splicing. Addition of nucleotides to the 3' and 5' we are talking about capping and poly A. And modification of certain nucleotides means methylation. Okay, so this is what they are saying here. Then we talked about our ribosomes. So ribosome is the largest and the most complex ribonucleoprotein. And what is a ribonucleoprotein? Ribonucleoprotein is nothing but RNA and protein complexes. And when we talk about ribosomes, what are ribosomes? Ribosomes are the protein biosynthetic machinery, and prokaryotic and eukaryotic ribosomes are different. Eukaryotic ribosome is a little more complex in structure. It has greater number of subunits. See, prokaryotic ribosome is known as 70S ribosome, and eukaryotic ribosome is known as 80S ribosome. This 70S ribosome has two subunits, 50S and 30S, and this 80S ribosome has two subunits, 60S and 40S. Point to remember that these subunits are not additive in nature. This S represents the sedimentation coefficient. Then we talked about the tRNA processing, how tRNA processing takes place. So this tRNA, the primary transcript of the tRNA has to be converted into a mature form of tRNA. So there are two sequential steps that converts the primary transcript of a tRNA into a mature tRNA. First is that specific endonucleases and exonucleases will cut this primary transcript. Okay, this is a general this thing modification, and then certain base are modified, and these base modifications are unique to each particular tRNA type. So this is how tRNA gets modified. Okay, this is the tRNA modification in the case of prokaryotes, and then in the case of tRNA modification in the eukaryotes, there is addition of 16 nucleotide 5 prime leader, 14 nucleotide intron is removed, 2 extra 3 prime nucleotides are added. All these things happens. Then RNA-sp, what is RNA-sp? RNA-sp is the ribozyme, which is a catalytic RNA. So whenever anybody asks you what is RNA-sp, you always say that RNA-sp is the catalytic RNA. Then we did some questions. Then we came to lecture 2. Lecture 2, we talked about the RNA processing. What are the different RNA processing events that takes place after the formation of your primary transcript? So this is a primary transcript. Either of the strand takes parts as a template for the transcription to take place. First, there is 5 prime capping, then there is 3 prime poly A tail addition, and then there is a splicing that happens. This is sequential: capping, tailing, and splicing. Okay. Then before going into splicing, we talked about HNRNP and SNRNP. Why we talked about HNRNP and SNRNP? Because they both are involved in the splicing process. SNRNP, in fact, are the component of the spliceosome machinery. And these SNRNP, how they are generated, we talked about. This SNRNP is required for the splicing. It helps in the splicing process. But how this SNRNP is generated? So this SNRNP, it is generated, it gets modified, it has a 5 prime cap. It goes to the cytosol. In the cytosol, its 5 prime cap, 5 prime cap gain 2 methyl groups. And then after the gaining of 2 methyl groups, it is again imported back to the nucleus where they function in splicing. Okay. So what is happening? First, this SNRNP is synthesized in the nucleus. It has a normal 5 prime cap. Okay, this SNRP synthesized in the nucleus gets modified, gains a normal 5 prime cap, and then it goes to the cytoplasm. And in the cytoplasm, it gains 2 methyl groups. This 5 prime gap gains 2 extra methyl groups, and after this gaining, it comes again to the nucleus, and then it helps in the splicing process. So SNRNP is required for splicing. HNRNP also required for splicing. How? It prevents the formation of any unwanted secondary structure. That means it keeps the RNA into the single stranded form. Okay. And then we talked about splicing. What is splicing? Splicing is the process of cutting the pre-mRNA to remove the introns and joining together of the exon. This is known as splicing. So whenever splicing takes place, I have already told you, we have discussed in lecture 3 in detail, that certain sequences are highly conserved in splicing and should be remembered. At the 5 prime end, it is GU. At the 3 prime end, it is AG. Okay. And branch point is A. Fine. Then we did some questions which are related to this second lecture. And then we talked about how this spliceosome complex works. So we talked about that a lariat structure is formed and your intron is removed as a lariat structure. See, first there is a cut here. This goes and binds at the branch point. And then there is a cut here and then it gets removed. Then we talked about 5 prime capping. What is 5 prime capping? 7 methyl guanosine is added and there is a 5 prime linkage. These 2 points which are highlighted are important. And how about the functions of this 5 prime cap? It helps the mRNA from degradation, protects it from degradation, increases the translational efficiency, helps in the transport to the cytoplasm and splicing of the first exon. Then we talked about how polyadenylation and takes place, how poly A tail is added. We talked that wherever there is a polyadenylation signal is there, approximately 20 nucleotides away from this, there is a cut, and then poly A polymerase adds the polyadenosines here. Then we talked about then we talked about methylation. That apart from capping, poly A tailing, and splicing, methylation also occurs, and this methylation is not only conserved, these are largely conserved in mature mRNA. This methylation is not only conserved, but also it makes your mRNA resistant to degradation. So therefore, mRNA methylation is again a very important modification that happens. Then we talked about alternative mRNA processing. Now, what is the meaning of alternative mRNA processing? Alternative mRNA processing means that same primary transcript which has been synthesized can give rise to more than one kind of mature mRNA. Okay, and same primary transcript can give rise to more than one type of mature mRNA. How it can happen? It can happen through many ways. It can happen through alternative splicing, taking alternative promoter sequences, or taking alternative poly A sites. So we have dealt in detail about the alternative this thing splicing, that depending on that which intron you are splicing, your exon joining would be different, and when your exon joining would be different, it will lead to the formation of different kinds of mature mRNA, and these mature mRNA will form different proteins. This is your alternative splicing. And then I talked about poly A tail, that the primary transcript can have more than one poly signal sites can be there. So depending upon which site has been used, the length of the mature mRNA would be different. Also, since the length would be different, the sequence would also be short or long, whatever it is, and therefore different kind of mature mRNA would be synthesized. Okay, and then we dealt with some questions. So lecture 4 was about RNA processing and life cycle. So this is what we have covered. So do you have any questions? Anybody has any questions related to this thing? So now what I have thought of doing is that as many lectures are there, there were 3 lectures supposed here. So first I just kind of summarize lecture 1, the important points, or if there's any concepts that I feel should be explained to you, I explain those concepts, and then we solve 2 3 questions which are related to lecture 1. Then we'll go to lecture 2. We'll just understand the theory of lecture 2. Lecture second, the subsequent lecture, we'll see the important points, we'll revise the topics, and then we'll solve some questions of lecture 2, and so on for all the lectures. So what happens that there is a balance between your theory portion also and covering the question portions also. Okay. So in this way, I mean, it would not be boring. Otherwise, it would be just theory. You might get bored because it's a 2-hour class. And I don't want to end the class early by just making you solve the questions, because I really want and I put my effort so that I can explain you something. And even if you're attending for 1 hour or 2 hours, even if you can attend even one topic, if you're able to attend, I mean, grasp and understand, your it is worth spending your time here, and it is worth spending my energy also here. Okay, so this is a very good opportunity for you students to come and interact, and if you have any doubts, you can ask me and you can clear your doubts here. So any questions, anybody? Anything that you want to say, any suggestion, anything, if you want to ask me, you can type in the message box, or you can directly ask me here by unmuting yourself as well. Any questions, any problems? Are you happy with the way we are going? Do you want me to continue in this way or not? I always ask you guys, because this everything is for you. Okay, I'm not getting any money, I'm not getting anything out of it. It is just for your benefit. Okay, fine then. We'll meet again in the next class. Again on Tuesday. The time is the same, 7 to 9. The recordings will also be available, will be made available to you, the whole recording, plus your presentation. This presentation will also be made available to you. I think you have a link or something from where you can go and see the presentation, I feel. So, okay then, we'll stop the class here. Okay then, bye everybody. We'll meet again next week, Tuesday. Okay.